So the biomolecules, right? All the elements present in a sample of earth crust are also present in a sample of living tissue. Now, the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organisms than in earth's crust. We use trichloroacetic acid. Okay, so one is called the filtrate, and more technically the acid soluble pool, and the second one is retentate or the acid insoluble fraction. So we found thousands of organic compounds in the acid soluble pool. Now nitrogen is very little in earth crust, and silicon is very negligible in human body. Baki hydrogen, carbon, oxygen, nitrogen, sulfur is more in human body. Then sodium, calcium, magnesium, silicon more in earth's crust. Now so all the carbon compounds that we get, all the carbon compounds that we get so all the carbon compounds that we get from living from 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 living carbon from living tissues can be called as biomolecules. However, living organisms have also got inorganic elements and compounds in them. A slightly different but destructive experiment has done. All the water evaporates, the remaining minerals gives dry weight. So all the carbon compounds are oxidized to gaseous form CO2 and water vapor. All the carbon compounds are oxidized to gaseous form, that is CO2 and water vapor, and the remaining is ash. So therefore elemental analysis gives elemental composition of living tissue in the form of hydrogen, oxygen, chlorine and carbon, while analysis for compounds gives an idea of the kind of organic and inorganic constituents present in living tissues. Now amino acids, these are organic compounds. These are substituted methanes. They contain hydrogen, hydrocarboxyl group, and amino group, and a variable group designated as R group. So based on the nature of R group, there are many amino acids, but proteins are only of 20 types. The R group in this proteinaceous amino acids could be hydrogen in case of glycine, and methyl group, that is alanine, and hydroxyl methyl, that is CH3OH, it is CH2OH, that is serine. Now the chemical and physical properties of amino acids are essentially of the amino NH2 carboxyl COOH and functional R group. So okay, now based on the number of amino and carboxyl and carboxyl groups, there are acidic, that is glutamic acid, basic, that is lysine, neutral, that is valine amino acids. So there are aromatic amino acids, tyrosine, phenylalanine, and tryptophan. A particular property of amino acid is the ionizable nature of NH2 COOH. Hence, in solutions of different pH, the structure of amino acid changes. So, in the second diagram, you see NH3 positive and COOH negative. It's in the ionous form. So, this is butyric ion. A NH3 positive, right? COOH is present. So, in C COO negative. So, NH2 present. So, this is it. Now, lipids are generally water insoluble. They could be simple fatty they could be simple fatty acids, like a fatty acid has a carboxyl group attached to an R group. A fatty acid has a carboxyl group attached to an R group. Now the R group could be methyl, ethyl, and higher number of CH2 groups, that is 1 to 19. Carboxyl group, that is COOH and R. So these are the fatty acids. Palmitic acid 60 carbons including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxyl group carbon. Fatty acids could be saturated without double bond and or unsaturated with one or more C double bond. Okay, so unsaturated and saturated fatty acids could be. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Many lipids have both glycerol and fatty acids. Here the fatty acids are esterified with glycerol. Here the fatty acids are found esterified with glycerol. Now they can be monoglycerides, diglycerides and triglycerides. Now these are called fats and oils based on the melting point. So oils have lower melting point. Example, gingerly oil, and hence it remains as oil in winter. So can you identify a fat from the market? Some lipids have phosphorus and a phosphorylated organic compound in them. These are phospholipids. Now they are found in cell membrane. So lecithin is one example. Some tissues, especially the neural tissue, have have lipids with more complex structures, right? So the nitrogenous bases are adenine, guanine, cytosine, uracil, and thymine. So when these nitrogen bases are attached to a sugar, these are nucleosides. And if phosphate group is esterified to the sugar, then nucleotides, adenosine, guanosine, thymidine, uridine, cytidine, and these are nucleosides. Adenylate acid, thymidylate acid, guanylate acid, uridylate acid, cytidylate acid, these are nucleotides. Now nucleic acids like DNA and RNA consist of only nucleotides only, right? So both DNA and RNA work as genetic material. C5H10O5, this is ribose, and C6H12O6, this is glucose. Remember the diagram: OH, OH, OH down in ribose, and then CH2OH, and OH at the third position. OH is on the top in glucose, and then OH is down at the fourth position, and CH2OH. So this is it. Then glyceralenine, serine. Then you have fatty acids, that is COOH and R group, that is palmitic acid. Then CH2OH, CHOH, CH2OH, this is glycerol. Then triglyceric acid. Here are R1, R2 and R3, these are fatty acids, right? Fatty acids are like R group, that can be methyl, ethyl, 1 to 9, 1 to 19 carbon, 1 to 19 carbon, and then 1 to 19 carbon, and then mm, okay, to 90 carbon, right? R1, R2 and R3. So this is it. Like you can see, there is a removal of OH H2O in the triglyceride. So this is how the structure is formed. Is kind of esterification. So R1, R2 and R3, these are the three fatty acids in triglyceride. Now phospholipid, it is lecithin. It contains two R1, R2, that is two fatty acids, and one phosphorus acid, that is H3PO3, I guess, H3PO4, and then choline. Okay, then nitrogen has, you see it's quaternary nitrogen because it attached to 4 3 CH3. Then fats and oils, these are lipids. Cholesterol, you see OH group is present. One heterocyclic ring is also present in cholesterol. So adenine, that is purine, then adenosines, and then adenylic acid, right? So adenine, that is two heterocyclic, urine has two heterocyclic ring, and pyrimidine has one heterocyclic ring, that is uracil. So then uridine, like uracil, OH. So this is it. Now primary and secondary metabolites. Amino acids and sugars, these are primary metabolites. Okay, uh, these are primary metabolites because they have an identifiable function. uh, okay, while primary metabolites have identifiable function and play known roles in normal physiological processes, but secondary metabolites like alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigments, scents, gums, spices, these are secondary metabolites. And and do not know the function in our body, so these are secondary metabolites. So many of them are useful: rubber, drugs, spices, scents, and pigments. But some secondary metabolites have ecological importance, right? Now, biomacromolecules. So biomacromolecules, right? Biomacromolecules, these are acid insoluble fraction, and biomicromolecules, these are acid soluble fraction. Now, uh, so the acid soluble, okay, 18 to 800 Dalton molecular weight. These are biomolecules. Okay. Then the acid insoluble fraction has only four types of organic compounds: proteins, nucleic acids, polysaccharides, and lipids. These classes of compounds, with the exception of lipids, have molecular weight in the range of 10,000 Daltons and above. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids whose molecular weights do not exceed 800 Dalton come under the acid soluble? Because lipids are indeed smaller molecular weight, but not only as such, but only arranged into structures like cell membrane and other membranes. Now when we grind this cell membrane and other, they form the vesicles. Now these are water insoluble, so this is why they remain membrane fragments in the form of vesicles separated along with the acid insoluble, and hence in the macromolecular fraction. But lipids are not strictly macromolecules because their molecular weight do not exceed more than 800 Dalton. Now the acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Okay. Now water is the most abundant chemical in living organisms. Now this table. Average composition of cells.